We're going to look at the processes of branch migration and resolution and the proteins that work together to carry out these reactions. So here's the long single-stranded tail made by RecBCD and then covered with RecA, and it invades the other duplex to give you the first junction. So remember that in the normal recombination intermediate for the double-strand break repair pathway, you have two of these. You have two long single-stranded tails that both need to get engaged with the other duplex in order to get the repair process. But for looking at these steps of branch migration and resolution, we're going to just focus in on looking at one branch at a time. And just remember that the intermediate made by the double-strand break repair pathway has two of these junctions that both need to go through these processes. So the first step we're going to look at is branch migration, where this can move to different positions on the DNA. And the second is the resolution of these crosses by holiday junction cleaving enzymes. And we'll look at those reactions partially separately, but you'll also see that in many organisms, the enzymes that catalyze these steps are actually functioning together in a super complex. So this is the way we draw branch migration when we are just looking at the sort of flat view of the double-strand break repair pathway. And so what branch migration is the movement of this branch to a different spot between these two double-strand pieces of DNA. Branch migration is the reaction of where you break hydrogen bonds in the double helix here and remake them in the new double helix, which is going to be the hybrid double helix, so these ones here. So this branch migration reaction, if you think about it, there's no real reason why that should require the input of energy. You're taking two molecules that are of the same energy. They have essentially identical Watson-Crick base pairs, so it shouldn't really require an input of energy to get that branch to migrate. And actually that's true, that spontaneous branch migration does occur. It can be a spontaneous reaction across DNA molecules like this. However, during the actual pathways of the double-strand break repair, it's important that these reactions be catalyzed for two reasons. First, you can imagine that if they're catalyzed, they can be sped up and occur much more rapidly, and that is in fact true. There's a motor protein that takes care of this process and makes it go much, much faster. And the second advantage of using proteins to carry out this step is it can ensure that the branch migration is unidirectional. So if it chooses to move it this direction, it won't go back. Whereas if you were looking at the spontaneous migration of this branch, it would take a few steps forward, a few steps back, and those are all going to be energetically the same state, and it won't have any reason to drive the reaction forward. Okay, so to look at the process of branch migration and holiday junction resolution, I want to go back to a structure we looked at a couple lessons ago where we've opened these junctions. We've taken from this view, which is common when people talk about recombination, to square view here, and then to the square planar view where you don't have any crossing strands. This is the easiest structure to observe to follow these processes of branch migration and resolution, and in fact it's the type of structure that some of the proteins are looking for. So here we have two parental duplexes, and we have coming out of this reaction, we want to make more of the newly paired double strands that are this one and this one. So how do the proteins work to catalyze the reaction of moving this branch and then eventually getting this branch cleaved? The first protein you need to know about for this process is the RuvA holiday junction binding protein. And it is a DNA binding protein that binds not to a specific sequence, but it's very structure specific. If you give it a holiday junction shaped DNA molecule, it will bind to that DNA molecule regardless of what the sequence is. It's really specifically looking for that structure. It's a tetrameric protein, so it's got one subunit to go between each of these 4 arms, and it actually holds the DNA in such a manner that it's almost completely base paired. There's just a couple nucleotides that aren't paired right in the middle of this structure. RuvA has this really important job of recognizing holiday junctions, holding them in the right configuration so that they can move, and recruiting by protein-protein interactions the second protein, the RuvB protein, to this holiday junction structure. So the RuvB protein is very similar to a class of proteins that we've already talked about. It's a hexameric motor protein, very similar in structure to the hexameric DNA helicases that work during DNA replication. So you learned about DNAB and the MCM proteins. RuvB is a member of that same protein family. However, it doesn't really function as a helicase. It functions just as a DNA translocase to help pump the DNA through this scaffold that is RuvA to promote the branch migration reaction. These are the parental duplexes, and these are the new hybrid duplexes. And RuvB, using the energy of ATP binding and hydrolysis, will pump the DNA through this structure, and at the same time, that will rapidly move the holiday junction through this complex. And it's a very efficient and powerful pair of proteins that can keep this branch migrating for tens to hundreds to even thousands of base pairs within a recombination intermediate.